Loratadine is a drug I know a lot about because I was Senior Director of Drug Metabolism at Schering-Plough during the period when we were developing the second generation drug to follow loratadine, namely desloratadine, also known as Clarinex. The FDA had concerns about the major metabolite of Clarinex, which was hard for us to address. And when I saw the results generated on the Hurel platform, I saw a tool that could have helped us greatly if only we had had it at that time. 3-hydroxydesloratadine is the major circulating metabolite in humans. The FDA was concerned about it because first, animals do not form the 3-hydroxy metabolite, and so were not exposed to it in the preclinical safety studies. And second, human formation of 3-hydroxy was found to be polymorphic, meaning that 7% of patients were slow metabolizers who received a 6-fold greater exposure. These concerns caused the FDA to require Schering to study 3-hydroxydesloratadine, a requirement that we had a very hard time fulfilling. Since animals don't form the 3-hydroxy metabolite, we had to turn to human in vitro models. We tried microsomes, S9 fractions, hepatocytes, and even express CYP enzymes, but they all failed. Trying to solve this problem of biologically generating the 3-hydroxy was a big part of why it took 44.8 months, about 4 full years, for the FDA to approve Schering's NDA for Clarinex. And what's more, it was the entire reason why the FDA imposed a subsequent requirement to continue to study the enzymology of 3-hydroxy formation even after the NDA was approved. Now I'm going to show you the loratadine metabolite generation data that was produced on the Hurel model. This work was performed by QPS, the Raleigh-based CRO, using co-culture plates provided to them by Hurel. So here's the data table. And as you can see in the left-hand column, the parent, loratadine. Next to it are the 6 metabolites that are observed in humans. And down at the second from the bottom, you can see the one we could never get, 3-hydroxydesloratadine. Now, if you look across the row, you can see why the FDA was concerned. 3-hydroxy is by far the major circulating metabolite. As you see on the right, the Hurel model was compared to the 2 industry standard in vitro models, microsomes and a conventional plated monoculture of primary hepatocytes. And as you see here, and just as we saw 4 years ago at Schering, the metabolite was not generated in either the microsomes or the hepatocytes. But the 3-hydroxy metabolite is generated in the Hurel model, the first time I have ever seen it generated in any in vitro system. What if we had the Hurel system to work with 15 years ago at Schering? We would have been alerted early in the preclinical development that there was a human metabolite not present in the safety studies. And more importantly, we would have been able to figure out what enzyme made this metabolite. And we would have been able to explain the slow metabolizer phenotype to the FDA. This would have reduced the long delay in approval of the Clarinex NDA and obviated the post-approval commitment that Schering study the enzymology of 3-hydroxylation. Claritin, the first generation drug, was $1 billion blockbuster for Schering-Plough. The second generation drug, Clarinex, was intended to be introduced 3 years prior to when it ultimately was. It's hard to assess the full economic cost of that 3-year delay in the launch of Clarinex, a delay which was caused in part by the lack of a correctly predictive in vitro tool. But it is certain that the Hurel system, had it been available, would have reduced that cost greatly.